Lulu. There an echo. So that we did the area flow steam. So cellular automata are a very nice type of system to use to explore the origin of life. We have been able to see a lot of different behavior and capacity emerge from a wide variety of cellular automata. And some capacities are also in the very crude to-do list to be alive, so such as self-constitution and maintenance or reproduction. For example, in earlier work on Lenia, we have seen creatures that seems to be able to move around and duplicate themselves and be able to keep their form. But in this kind of cellular automata, we don't really have the notion of evolution. Notably because the rule stays the same during all the simulation, and same pattern won't produce different results depending on some kind of genome or something like. So in our participation, we used the cellular automata model that was not really new. So I reused it during my master internship. So it's Lenia, which was quickly mentioned in the previous presentation. This is a continuous cellular automaton, which has the nice properties of conserving mass, so the amount of activation in the whole simulation, and also enables evolutionary dynamics. So in this model, the activation of the cellular automaton is representing basically mass. So the brighter the cell, the more matter there is in it. And we kind of simulate particles that move according to a flow map. And the next state of the cell is defined by the movement of particles that are in its neighboring cells and that come into the cell to form the new state. The matter move, and then we diffuse it to have a kind of nice properties of not too much concentration. Yeah. Small video that show how it's done. So with this formalism, we have matters move around. And what is also in the original paper is the idea of embedding the rules of the world inside each cell, so that each cell correspond to different rules, and the world is not uniform anymore. So in the next simulation, and the one I show in the video, I color each part of the space with its with a color. Each color corresponds to one set of physical rules that apply in this part of the world, and the brightness is like the mass. And from there, what we would like to see is an emergent evolutionary process, because it's not directly creatures with a genome, but it's rules that move around, propagate. And what we call a creature is made of an ensemble set of cells neighboring cells. And the goal of this work is to create an evolutionary process with an unbounded growth of complexity over the course of a single simulation. It's not like in earlier Lenia works in which we make creature evolve by creating new simulation with new rules for the updates, but it's inside a single run. We want an evolution process. And we see that at the very beginning of this simulation I'm running now, we had some very crude dynamics, but at the end, so it takes a lot of time to simulate, but we can see the appearance of rules that enables, for example, gliders and a lot of small scale dynamics. But yeah, in other simulation, we can see also larger patterns. The way the parameters and the rules of the world propagates is actually very important. There is no real fitness function in this evolutionary process. It's an intrinsic fitness function that will actually be linked to how we propagate the parameters inside the simulation. For example, if the new parameter of the cell are the average of its table, like on the left, we will have some kind of uniformization, and yeah, the dynamics will be in the end the same everywhere. If we optimize, for example, when parameters arrive with matter and we choose the one that has the most kinetic energy, which is something we could think of being more fit, then the simulation just breaks because we only have like high-speed matter that is moving, and only the parameters that are linked to that are selected. Another one with the parameters are linked to the amount of matter accompanying them, which actually led to good, quite interesting dynamics, but which we don't adopt in the end. And each of these mixing rules will definitely end up having a different profile in the evolutionary dynamics that will be observed. In the participation, so it's my contribution during my internship, I try to modify these mixing rules to parameterize them, having some more general description, and notably by decoupling the actual movement of matter and the spreading of the mixing rules of the local rules of the dynamics. Here are some examples that were obtained by running simulation with this kind of rules. We end up with a very large variety of patterns, like some are big. Remember that the colors are associated to the parameter and dynamics of the cellular automaton. So we have organisms that have different parameter and dynamics that create different parts of the larger organism. Here we have some kind of membrane and nucleus, and some merging and division of the patterns, which all appear in the single simulation. So finally, now that we have a parameterized way of creating evolutionary processes, what we are doing is trying to explore the space of evolutionary processes, not only trying to explore for interesting patterns, but for interesting evolutionary dynamics. The tricky part with this kind of work is that the way to measure property of evolutionary processes in the case of continuous error automata are not quite ideal yet. However, by combining multiple metrics, such as one for evolutions, one for complexity or novelty, we can use some algorithm of automated discovery to try to explore and sample this space. So yeah, thank you for listening, and don't forget to check the Lenia original paper, which is did most of the work for this submission.
Thank you.